It seems nobody has come offline. Then I will start my class. So today we will talk about carbohydrates. This is the third chapter in my portion. And then I'll have lipids. After carbohydrates, we will, in the next class, we will do lipids. And then we will go to cell biology and molecular genetics, that particular part. So, what do you people understand by carbohydrates? Can anybody tell? What do you understand by carbohydrates?
Sir, a biomolecule which consists of carbon, oxygen, and hydrogen.
So, any other biomolecules which consists of carbon, hydrogen, and oxygen? Do you know? Basically, most of the biomolecules are composed of carbon, hydrogen, and oxygen. Some of them have even nitrogen. So, that is exactly not the definition of carbohydrates. It is the definition of carbohydrates is hydrates of carbon. What is a hydrate? H2O, water. Carbon is attached to water. It's a hydrate. It is watered compound. Okay. So
Yes, sir.
whenever you, so I'll share my the screen, and you people must tell me if it's visible or not. Is it visible?
Yes, sir.
Yes,
Yes,
sir.
sir.
So car means carbohydrates, and basically you can say CH2O and N. So basically the word is hydrates. Even lipids also have got oxygen, but less. So many of these are biosynthesized inside the living body from simpler processes by process of gluconeogenesis and photosynthesis. So a carbohydrate basically is synthesized in living systems through the process of gluconeogenesis. That is the synthesis of carbohydrates from non-carbohydrate sources. And you also have another thing which is important, that is photosynthesis. The main way. No photosynthesis, no life. So carbohydrates can be divided into, first is the monosaccharide, which has got one sugar molecule. That saccharide, SAC, saccharide, this comes from the word shakar. So one sugar molecule is monosaccharides. And they are like glucose, fructose, galactose. Then you have the disaccharides, where you have two sugar molecules are attached. For example, sucrose, lactose, and maltose. Then you have the oligosaccharides, where you have about 2 to 10 sugar molecules attached. Example, raffinose and stachyose. You also finally have the polysaccharides, 10 or more sugars molecules. Most important among them are starch, glycogen, and cellulose. So starch and glycogen, they are sometimes called as storage polysaccharides. Storage polysaccharides. And cellulose and chitin. Chitin is another polysaccharide. They are known as structural polysaccharide. Structural. Now let us look at the structure of a molecule known as glucose. This is glucose here. So monosaccharide, they are among the monosaccharides. They are nothing but simple sugars are aldehyde and ketone derivatives of straight chain polyhydroxyl alcohol. Hydroxyl group is OH. So if you look at this structure, you will see many OHs. 1, 2, 3, 4, 5. So basically polyhydroxy, so that many hydroxyl groups are there. And they are either, this is an aldehyde group, polyhydroxy compounds, aldehyde or ketone derivatives. It can be also ketone. We will see in the case of fructose. So in the case of glucose, you will see that the first carbon is a carbonyl carbon, or which contains the aldehyde group. They contain the aldehyde group. And then you have the second carbon, third carbon, fourth carbon, fifth carbon, sixth carbon. Sixth carbon is CH2OH. The third carbon in glucose, this is essentially glucose. You will see that the OH is on the left hand side. So this is known as the Fischer projection, and it is known as D-glucose. Now, you will see that there is carbon 1, 2, 3, 4, 5, and 6. Now you have to find out which is the chiral carbon. So a chiral carbon is a carbon which is attached to 4 different groups. Chiral carbon attached to 4 different groups. Attached to 4 different groups. Now if you see this, the first carbon, it is not attached to 4 different groups. You have this as one group. You have the second group, and this is the third group. So this is not chiral. But the second carbon, you have it is attached to the carbonyl first. It is attached to an OH. It is attached to this H, and it is attached to another different group. So this is a chiral carbon. Can you say if the third carbon is the chiral carbon? বলতে পারো তোমরা third
Yes,
carbon?
sir.
Okay. Why is it chiral carbon?
Sir, it is attached to 4 different groups.
Very good. 4 different groups. This is the first group, second group, third group, and fourth group. Similarly, the fourth is also a chiral carbon, and the fifth is also a chiral carbon. But the sixth is not a chiral carbon. This carbon is attached to 2 hydrogen atoms. So it is basically attached to one big group, and then it's attached to an OH and then 2H. So it is not a chiral carbon. So in a 6 compound glucose, there are only 4 chiral carbons. And depending on where the OH is in the last chiral carbon, depending on where OH is in the last chiral carbon, the carbohydrate is classified as D glucose or L glucose. D is capital, L is capital. This is based on the structure of glyceraldehyde. In glyceraldehyde, if the chiral carbon, there is only one chiral carbon in glyceraldehyde. There is 3 carbons, but one chiral carbon in the center. If the OH is on the right hand side, it is D. If it OH is on the left, then it is L. This is not the dextro-levo, small d, small l. Small d, small l is when you in a solution, when you pass a polarized light, the light either turns to the left or the light either it turns to the left, right, after it hits this carbon. That is optical rotation. But this is not from optical rotation. It's based on whether the OH is on the right or whether the last chiral carbon OH is on the left. I hope it is clear.
Yes, sir.
Very good. So now we have come to fructose. In glucose and fructose, everything is same. This is the first carbon, this is the second carbon, third carbon, fourth carbon, fifth carbon, sixth carbon. It is the same, only that it has got a keto group. This part is the keto group. This is a keto group, not an aldehyde group. But the third OH is on the left hand side, the fourth OH is on the right hand side, fifth on the right, and therefore this is D-fructose. So in nature, D sugars are the most commonly occurring compounds, or the sugar form is always D sugar. Whatever is found in your body, the sugars, they are mostly in D. There are one or two exceptions of L, but mostly they are all D. And in amino acid, it's just the other way around. You don't have D amino acids, you have L amino acids in nature. So D glucose and L glucose, they are mirror images of each others and are called as optical isomers or enantiomers. This is D galactose and this is D glucose we are talking about. So D glucose and D L glucose, they are mirror images. So if you stand in your mirror, in front of a mirror, your left hand will come to the right. Does it come like that? Left hand on the right when you stand in front of the mirror?
Yes, sir.
No, it does not. তুমি বাঁ হাত তুললে তুমি বাঁ দিকেই মিররে তোমার বাঁ হাত দেখবে. So when you take a selfie, if you don't flip it already, you will see it like that. But in a say for example on your face, but if you place a molecule and place a mirror like this, this is a mirror image. Is it clear?
Yes, sir.
If you stand in front of a mirror looking, admiring your face, if you close your left eye, you will see your left eye only closing. Right there. Am I clear?
Yes, sir.
But if you put your hand in front of the mirror, you will see that both your hands are colliding in front of each other. It is just not a duplicate copy, it's just the opposite. So when you are writing, sometimes during this lockdown, people started to use these glass boards for teaching classes, for making videos for classes. So they used to write on the glass board and they used to take a video from the other side and they used to flip rotate the video. So the moment when you used to see the video, you would see that this person is writing through his left hand. It's no problem. But actually he's writing from his right hand, just to keep for students who are viewing it, they can see it in the correct orientation. So this was one way of teaching in this lockdown time. But that Glassboard, from glassboard, this is much better. So these are known as enantiomers or optical isomers. In galactose, which has one OH group to the left and one OH group, the fourth one, and this OH group is in the glucose, right? The others, the third carbon is the same. They are also known as epimers. They are also enantiomers. All epimers are enantiomers. But glucose and fructose, they have the same molecular formula, but their structure totally is different. The carbonyl and keto group, and they are known as isomers. Am I clear? Can you do you know what now? Can you define enantiomers, epimers, and isomers if given in the exam? Yes, no?
Yes, sir.
Okay. Now we will go to the cyclization of glucose. This is also from Fisher to Haworth projection. From Fisher to Haworth projection. So you have an alcohol ROH group and you have an aldehyde group. This group is different from this group. R and R dash. This is R and R dash. So when they fuse, this double bond, it breaks and another H is attached. This double bond, it breaks and another H, it becomes OH group. And this double bond becomes a single group. And they attach with each other. This ROH comes so attached to this carbon here. And this R attaches over here. So it forms the compound known as hemiacetal. So here there are two different compounds. Here there are two different compounds. Okay. There are two different compounds over here. But when the attack is from the same compound, this is glucose in linear form. Aldehyde group 2, 3, 4, 5. This is glucose in linear form. I've just taken your attendance. Just hold on for 1 minute. Your attendance has come. MSC, same one. Today's date is 03/11/2023. Attendance. Okay. Let's get back to work. So this is D-glucose in linear form. Now, this aldehyde group, it is attacking the fifth alcohol over here. Now you are just simply turning the molecule. এটাকে এই সোজা ছিল, সোজাটাকে এরকম করে বেঁকালে, you are just twisting it. They become each to it. This OH and this aldehyde group will attack each other, and the water molecule will go out, and you have this cyclic structure. This structure is known as a pyrene ring. This is known as a pyrene ring or pyranose, and this is known as beta D glucose Howard projection. And this first carbon which is over here, this carbon is not written. These dots are in the carbon. Here the carbon is written. In number 6, the carbon is written. The rest, in this part, this part, this part, the carbons are not written. So this first carbon, first is anomeric carbon. The first carbon is also known as anomeric carbon. That is because you see this OH is on the top. So when this breakup happens, when the OH is on the top, then it is beta. But when the OH is below and H is on the top, on the first carbon, that is known as alpha pyranose. And this is, we come to another phenomenon known as mutarotation. So this alpha glucose and beta glucose, they are anomers of each other. Anomers of each other. So in alpha glucose, the OH is below. In beta glucose, the OH is on the top. The rest remains the same. Now, when you give them, they may be diastereomers, but they have different physical and chemical properties. They have different physical and chemical properties. So optical rotation of alpha D glucose is 112.2 degrees. So when the polarized light goes on the plus direction, it moves on the right to plus 112 degrees. But beta glucose, D glucose, it moves 18.7 degrees. Okay, so they have different properties also. For example, if you have a disaccharide with beta glucose, you cannot digest it. Or plant cellulose, you cannot digest it. You cannot digest the beta glucose, the bond made from beta glucose. But you can digest alpha glucose, that is maltose. So when either of these pure substances is dissolved in water, the specific optical rotation of the solution slowly changes until it reaches an equilibrium value of plus 52.7. You put alpha in a solution, you dissolve it. First, immediate 112 degrees, then immediately it will go to 18.7. It will get converted into beta glucose. It will become 18.7. Again, it will come back to 112. So when you measure, you will get a reading of plus 52.7. So this alpha glucose in solution, it becomes a straight chain, the Howard projection, and then again it cyclizes and forms beta glucose. So this phenomenon is known as mutarotation. But in equilibrium, 63.6% beta anomer is formed and 36.4% alpha anomer is formed. Are you people clear about mutarotation?
Yes, sir.
Okay. Next is another process known as tautomerization. Another phenomenon known as tautomerization. This is the shifting of an hydrogen atom from one carbon atom to another to produce a compound known as enediol, or it is known as tautomerization or enolization. This enediol is a very important compound. This is the structure of glucose. Instead of drawing the aldehyde group like this, I have made it straight. This is D-glucose. Beside it, I have drawn fructose, D-fructose. Now, in solution, in alkaline solution, what happens is this double bond can migrate over here and this OH can come over here. and it forms an intermediate compound which has got a structure like this. And this is a temporary unstable form, and it is known as enediol. Remember this. This is also important. You need it in when you study glycolysis. This enediol intermediate is formed. Then this double bond can migrate back over here. And then if it migrates over here, this H will attach and this will become a keto form, and that is D-fructose. Again, that double bond can migrate back and go front. And what can happen is that this OH was on the right side. This OH can come on the left side. And that is a sugar known as D-mannose. And this interchanging in alkaline solution of D-glucose, this is known as tautomerization. It goes through an enediol intermediate. Are we clear here?
Yes, sir.
Okay. Now we come to a glycosidic bond. This is an alpha glucose. Alpha glucose, this is another alpha glucose molecule. This is the carbon one. I have not drawn this H on the top. I have not drawn this over here. That is, assume that there is an H over there. And I've not drawn the H, only the OH groups are drawn here. This is a very short way of remembering. This C1 and this C4, there is a condensation reaction and a molecule of water is eliminated. And the bond which is formed is known as 1 to 4, C1 to C4, alpha glycosidic bond. Because the C1 OH was alpha. C1 OH or anomeric carbon was in alpha anomer. Therefore, it is known as alpha glycosidic bond. If it was on the top, OH was on the top, and it would form a compound like this, then this would be known as beta glyco 1 to 4 glycosidic bond. And this compound which is formed is known as maltose. You can digest maltose. We had a drink known as Maltova. It used to give lots of energy because they were these disaccharides. They broke and gave lots of glucose, which gave you lots of energy. Maltova, because made from maltose. But if this was on the from the top of the glycosidic bond linkage was like this would be known as cellobiose, and you cannot digest this beta 1 to 4 beta glycosidic linkage is not digestible. So this compound which is formed, this is known as disaccharides. Similarly, this is a fructose. In glucose, this is known as a pyranose ring. In fructose, the same, since it's ketone, it is known as furanose ring. Okay. Here, the CH2OH is over here. The keto group is over here. The same thing, like the aldehyde for it has cyclized, we have cyclized it. And this is the beta. This is the beta configuration. And this is the alpha. So with sucrose, you will have a 1 alpha to 2 beta glycosidic bonds. Similarly, lactose, which is a combination of galactose and glucose, you have a 1 to 4 beta glycosidic linkage because this anomeric carbon is in beta. And This disaccharide is maltose. Alpha 1 to 4 glycosidic linkage. Glycosidic linkage. So far clear? Yes, no?
Yes, sir.
Then we go to the polysaccharides. Polysaccharides can be many, more than 10 units of sugar molecules attached. They can be structural polysaccharides, example cellulose and chitin. They can be storage polysaccharides, example starch and glycogen, or they can be a special group of polysaccharides known as GAGs or glycosaminoglycans. They are basically lubricants of eye, knees, lubricants. So the first one we let us study is chitin. It is found in crustaceans. Chitin is found in fungi. Chitin is found in crabs, the crustaceans, insect wings. They are made up of chitin. So basically, you have an A instead of the OH over here in the second carbon, you have an N-acetyl. You have a glucosamine group over here, N-acetyl glucosamine group over here. So this particular amine group over here, acetylamine. This is basically acetylamine amine group, and this acetylamine group, when they are bound like this is there are n number of units, one by one, but they are attached together. But instead of to this glucose unit, instead of OH, you have an acetylamine group over here, and this becomes a chitin molecule. This is the primary structure of chitin. And the famous cellulose. This is a beta 1 to 4 glycosidic linkage. This is beta. OH was on the top. OH. This is beta glucose. Many beta glucoses joined together. It forms cellulose. You cannot digest this. This is fiber. It will come out with your potty because you cannot digest it. But this fiber is very important for maintaining your digestive health. But cows and ruminant animals, they ferment it in their chambers. So out of the 4 chambers, in the they ferment it and they break them into glucose and small glucose units, and then they digest it. But we humans can't. We can only digest maltose. This is cellulose. But when this structure in the, now we go to a storage polysaccharide. First is starch. Starch is also known as amylopectin. So you have all alpha glucose attached together. They are all 1 to 4 alpha 1 to 4 glycosidic linkage, and in between there are branch points which are alpha 1 to 6. This is the 6th carbon. So the 6th carbon is attaching with the 1st carbon. So this is an alpha 1 to 6 branch point. So basically the structure is going like this, then it branches, then again it grows like this, again it branches, again it goes like this. So what happens is that many glucose units are stacked in a compact space. So when you eat rice or potato, which contains a lot of starch, they are broken down. These alpha, they are broken down. So there are enzymes which digest your salivary amylase. It breaks down alpha 1 in your mouth. The sweets which you take, sucrose, it's broken down into glucose and fructose. Salivary amylase. Then you have pancreatic amylase. Then there are branch point enzymes which break 1 to 6 glycosidic linkages. So this is storage polysaccharides. Now we come to glycogen. Glycogen has got the same structure as that of starch, but the branch points are much more. Every 8 to 14 glucose residues, there is a branch point. In starch, something around 25. So otherwise, the similar glycogen is stored in your skeletal muscle and your liver. Then you come to the next polysaccharide, the glycosaminoglycans. They are basically lubricants, or they are also known as mucopolysaccharides. Hyaluronic acid. You have heard of this term several times. Basically, you have the instead of CH2OH, you have COO minus. This is known as B-glucuronate, and the beta 1 to 3 glycosidic lipids, and this is an N-acetyl glucosamine. So you have got an acetylamine group attached to the glucose, and many units of this, and this causes a lubricant nature. So this is hyaluronic acid. You have heparin, blood thinner. So many negative charges over here. So this makes it very difficult for the blood to clot because of this negative charge, heparin sulfate. Now we come to glycoproteins. So they are carbohydrates attached with proteins. So most eukaryotic proteins are glycoproteins. They are associated with carbohydrates. You have the proteoglycans. If you look at the structure, it's a bottle brush structure. So these are proteins. This is a protein chain which we have drawn. On the top of it, here the carbohydrate chain is coming. This is the carbohydrate. This is a protein chain. On the protein chain are attached core protein chain are attached the glycan or the carbohydrate group. So it's almost like a bottle brush structure. Like a bottle brush structure, the brush with which you clean bottles inside. The next you come across is peptidoglycan. Most famous are your NAG and NAM. N-acetyl glucosamine, N-acetyl muramic acid. In bacterial membrane, this is very important. So this NAG-NAM, this is N-acetyl glucosamine attached to N-acetyl muramic acid. Now this is attached to amino acids lactin, L-alanine, isoglutamate, L-lysine, D-alanine. So such structures, this is a peptidoglycan. So it's like this is a peptide. It is attached to a glucose. So they are known as peptidoglycans. So this is the end of carbohydrate chapter. Some people like me, I cannot digest milk sugar, lactose. I immediately have problems, gas, flatulence, and stomach problems. So for these people, it is advised to take curd, where lactose is fermented to by lactobacillus, they make it into lactic acid. That you can easily absorb. So curd is very good because it contains lactobacillus. This is good for keeping your gut content very nice. So is it clear? Do people have any questions?
No, sir.
Okay, thank you then. Bye-bye.
Thank you, sir.
Thank you, sir.
Thank you, sir.
You're welcome.